Welcome and thank you for joining us for the fifth Agro Lunch and Learn webinar of 2024. This webinar series is only possible through the generous support from Eurofins Agroscience Services. We thank Eurofins very much for their continuing sponsorship. My name is Solito Sumalong, the Director of Regulatory and Product Development Services at AgriAuthority based in Kansas City. I'm an incoming executive committee member of the Agro Division of the American Chemical Society and a board member of the ISAA Agrochemical Network. I'll be serving as a moderator for today's webinar. Attendees like you should use the Q&A tool to submit their questions and comments, which will be answered at the end of the presentation. Before I introduce our speaker, I'll highlight the benefits of becoming an Agro member. Agro pays enormous benefits by enhancing your science, building your scientific network, and supporting your professional development. Please add the Agro division to your ACS membership or go to agrodiv.org and click on Become a Member to become an affiliate for $20 a year, regardless of your membership status with the American Chemical Society. I would also like to inform you about the next ACS Fall National Meeting in Washington, D.C. next August. If you have any questions about programming, contact our program chair, Dr. Ming Ma. The Agro Division is also active in executing the strategic plan of engaging new and existing members, delivering information through new means of communication, and creating a future-ready resource for the global agrochemical community. Kevin, Edmund, Leah, all there, and the Agro Division do need your help executing on this plan. So please see agrodiv.org for more details. The next ISAA symposium is scheduled for next spring, 2025, in Rio de Janeiro at the Windsor Bar Hotel from April 6th to 11th. The board of the ISAA Agrochemical Network will host Indorama Ventures, Isabella's company, our speaker, and over 450 global professionals in industry and academia like you for the best international symposium dedicated to agrochemical formulations, their co-formulants and adjuvants. This will be our second symposium held in Brazil and our first there since 2013. For more information, use the QR code on your screen or head to isaanetwork.org. Now I'm proud to introduce today's speaker. Isabella Kitano is a research scientist at the Crop Solutions division of Indorama Ventures in Sao Paulo, Brazil, and currently leads the BioSolutions platform. She holds both bachelor's and master's degrees in science in biotechnology from the Universidade Federal do São Carlos. She has more than 6 years of experience in biopesticides development and bioprospecting of new microorganisms, and today has a more focused orientation towards biopesticide formulation. Her webinar is entitled Overcoming Formulation Challenges, Navigating the Development of Biologicals. And now, Isabella. Boa tarde, Isabella.
Boa tarde. Thank you for the introduction. Let me share my screen. So thank you very much for the introduction and the opportunity to be here speaking to you all. So as Solito said, today I'll be discussing some of the key challenges in the formulation of biopesticides. So I took this opportunity to build a more technical presentation to allow us to delve deeply into the specifics of each active ingredient, their main challenges, and how the chemistry can help deliver these biological active ingredients. But before we go into the technical part, I just would like to introduce Indorama Ventures because we've been through some changes in the past few years. So Indorama Ventures is a global chemical company with a strong focus on sustainability and innovation. The company has these three main divisions, these three global business units: PET, fibers, and Indovinya, where I am. We are one of the largest producers of polyethylene terephthalate, the PET. So 1 in 5 PET bottles in the world are made from our resins. We are also one of the largest producers of fibers like polyester fibers, yarns, nylon, etc. And all of these can be applied to like premium baby diapers, manufacturing of airbags, for example. And finally, we have the most recent division, which we call Indovinya. So in 2020, Indorama Ventures acquired the American Huntsman Performance Products, and in 2022 acquired the Brazilian Oxiteno, and both of which brought with them a wealth of expertise in surfactants, solvents, and formulation technologies. And these acquisitions strengthened our capabilities, especially in the development of specialty chemistries. So basically, Indorama Ventures is everywhere. So now we are the leading surfactants and ethylene oxide producer in the Americas and the second ethoxylation company in the world. And all these surfactants, all these molecules can be addressed to different markets. And obviously today I'm gonna focus on the crop solutions market, especially the biopesticides one. I'm sure I don't need to emphasize how much the biologicals market has expanded in recent years. It's one of the fastest growing sectors, both in size and technical advancements. One of the key market drivers is undoubtedly the growing demand for organic food, increasing environmental awareness, and the need to expand the toolbox available to agriculture. I always say that biologicals will never completely replace chemical products because what really matters is finding the right balance between these different tools that compose the integrated pest management approach. And to find this balance to maximize productivity while minimizing environmental impact. And another interesting point is that biologicals are gaining increasing market penetration in row crops, particularly in Brazil, where we see significant adoption of biologicals in soybean production. Now, this image may be familiar to many of you. So in agriculture, biological products can be divided into several different categories. But what they all share in common is that their active ingredient are naturally derived, and they can have both biocontrol or biostimulant properties. Here we can see the range of active ingredients available today. As technology advances, we are discovering new molecules and organisms with useful properties for agriculture. And currently, the most products are based on microorganisms, particularly fungi and bacteria. However, we are now seeing the emergence of peptides and RNA molecules, which offer different mechanisms for pest control. And of course, the use of these technologies is expected to grow significantly as they work in synergy with other biopesticides. Here I've highlighted some of the main challenges when we are formulating these biological active ingredients, and I will comment on that, but I will address each one throughout the presentation. But when it comes to formulating biopesticides, we face a unique set of challenges that differ significantly from those encountered with conventional chemical pesticides. So one of the primary concerns is obviously stability. So ensuring the viability of microbial active ingredients, the integrity of peptides, and the stability of the RNA molecule are critical for maintaining the efficacy of the active ingredient. So microorganisms can lose their viability over time due to environmental stressors, while peptides and dsRNAs are prone to degradation from factors like temperature, UV exposure, and enzymatic activity. Compatibility with surfactants and other co-formulants also presents significant hurdles. While surfactants are necessary for improving dispersion, emulsification, and wetting properties, they also can be harsh on microbial cells or destabilize sensitive molecules like peptides and RNA molecules. So achieving the right balance between functionality and biocompatibility is essential to avoid compromising the product's effectiveness. Another critical factor is water activity. So we all know that water activity directly impacts microbial stability. So high water activity can promote microbial growth or lead to premature germination of fungal spores, while too low water activity may cause desiccation stress, reducing microbial viability. So controlling water activity is key to keeping the active ingredient in a dormant state, yet stable throughout the product's shelf life. The rapid assessment of shelf life stability is still a challenge also. We do not have a method to predict the stability of microbes in an accelerated way, like the chemical pesticides do. So we must perform the real-time shelf life at the appropriate temperature to assess this information, and this could delay the product development. Another challenge we encounter when working with mainly with fungi-based products is the hydrophobicity of fungal spores. So the water-repellent nature of spores makes them resistant to wetting, and this can result in difficulties during mixing in the spray tank or an uneven distribution during application. And this can potentially reduce the field efficacy of the fungi. And lastly, we must consider when formulating that environmental effects can be harmful to microorganisms, to the active ingredient. So factors like temperature fluctuations, UV exposure, and soil or plant enzymes can degrade the active ingredients or reduce their efficacy in the field. So designing formulations that protect the microbes and the other molecules against these environmental challenges is vital to ensure consistent performance under various conditions. Now I'll begin to discuss on each active ingredient. I won't be able to cover all of them, like peptides and RNA. So we choose to bring here the microbial ones as they are the most used and applied nowadays. So the first step when we are formulating a new active ingredient is to deeply understand it. While this may seem obvious, it's critical to really understand the key characteristics of the genus, the species, and especially the specific strain that you are working with. So you must ask if it's a gram-positive or gram-negative bacteria, if it's a fungi, which resistance structures do you have in your active ingredient. If it's aerial spores, submerged spores, blastospores, we have a wide range of structures. So it is really important for you to know what's inside of your active ingredient. And we know that strains, even within the same species, they have unique traits. So depending on where the strain was isolated, it may produce different molecules or exhibit varying levels of stability. Another aspect that highly impacts the formulation is the bioprocess, upstream and downstream. So the upstream process is the initial stage where the cells are grown, aiming to produce the biomass. It is the fermentation process itself. And we have mainly two types. So we have the solid state one, which is more applied to fungi, and the submerged liquid one, which is more applied to bacteria. However, we are now seeing a growing trend in liquid fermentation being applied to fungi as well. And the downstream process is any step of recovery, concentration, stabilization, or purification of this biomass, like drying processes after the upstream process. So now we can see some active ingredients after a drying process. So each step can affect the final characteristics of the active ingredient. It can affect the hydrophobicity of the active ingredient, the water content, and even the stability of the microorganisms. And it is also important to be aware of any interesting metabolite that the microbe might be producing, and you don't want to lose it during the processes. Bacillus, they are the most common biopesticides. It produces structures called endospores. It is widely known in the literature. And these endospores are highly differentiated cells that are extremely resistant to heat, harsh chemicals, and radiation. And they function as survival structures and enable the organism to endure unfavorable growth conditions, including extremes of temperature, drying, or nutrient depletion. So this sporulation process is mainly triggered when, for example, during the upstream process, a key nutrient such as carbon or nitrogen sources becomes limiting. And that's one of the main reasons why bacillus tend to be easier to formulate and to achieve a longer shelf life. On the other hand, we have gram-negative bacteria, which we have in this group microorganisms like Pseudomonas, Bradyrhizobium, Azospirillum, etc. And the gram-negative bacteria, they cannot form these persistence forms. They remain in the vegetative cell stage. So this is one of the factors that affect the storage stability issues that we have regarding gram-negative bacteria. And additionally to the fact that they cannot form endospores, gram-negative cell wall has a different structure from gram-positive ones. So here we have the gram-negative and the gram-positive wall. And their cell wall consists of at least two layers of lipopolysaccharide membrane, whereas the gram-positive cell wall is typically much thicker and consists primarily of a single type of molecule, which is called peptidoglycan. So around 90% of the gram-positive cell wall is composed of peptidoglycan. And in gram-positive and in gram-negative bacteria, only a small portion, a small amount of the total cell wall consists of this molecule. So That's why it is way more difficult to formulate these types of microbes and why they are more sensitive to some chemistries. There are some strategies that I've seen being used to prolong the shelf life, and one of them is using a carbon source, usually a sugar, to maintain the energy source, allowing the cells, the gram-negative cells, to maintain the basic metabolic functions while in a reduced metabolic state, in the formulation. And obviously, in a formulation, the cell's metabolism is not the same as in the fermentative process. But even at low metabolic activity, the cells need presence of a carbon source um, to maintain some basic processes to maintain so like membrane integrity, repair cellular damage, so maintain these basic survival processes. So theoretically, the presence of a carbon source would ensure that Pseudomonas can carry out these maintenance functions, reducing cell death during storage. And this sugar could also do a stabilization of the osmotic balance within the formulation. So this is a strategy that I've seen in the literature. Another common strategy that is applied is obviously the storage at low temperatures in order to slow down the metabolic processes. But we know that this strategy is costly and inconvenient, but it works. So it's a strategy that it's applied. And another strategy that I've seen is spray drying processes, spray drying the biomass. I don't have the data, but I think that there might be huge losses in microbial viability during these drying processes. So I don't know if this is the best alternative, and it is also very costly. And many industries they don't have a spray dryer to make this kind of processing. So I think that achieving a cost-effective formulation for gram-negative bacteria still remains a significant challenge, and we have been working on this. Another very interesting microorganism that have been explored as biocontrol agent is Streptomyces. They are gram-positive bacteria. They are well known for producing a wide range of secondary metabolites, including antibiotics, antifungals, and enzymes. So Streptomyces, they are unique. We can say that Streptomyces resembles a little bit fungi in certain ways, particularly in its growth and morphology. They have this dusty appearance. They form branching and filamentous structures that are also called hyphae, similar to fungi. And they also produce spores, much like many fungi do. But their spores are pretty different from endospores or fungi spores. I think they are like in the middle of them, but I think that submerged spores from Streptomyces is a challenge because we need to stabilize this spore in an environment with water, and water can trigger a premature germination. So working with this water activity balance is key to have a good shelf life. Finally, now moving on to fungi. Fungal spores are generally less robust than bacterial endospores. We all know that. Most fungal spores are produced either through solid state fermentation or submerged fermentation, and it can be followed or not by a drying process. In formulation development, our goal is to preserve these spores, these structures, in their dormant state, because we want to avoid triggering germination within the formulation and ensure it only occurs during dilution in the spray tank or once applied in the field. And fungal spores are particularly sensitive to water, which is why many formulations on the market are either oil-based or in the form of wettable powders. And one interesting movement that we can see is that we are discovering new liquid carriers that can carry this conidia without allowing them to have a longer shelf life without being harmful to them, without being harmful to their structure. Fungal spores are highly hydrophobic, primarily due to the presence of hydrophobins, which are small cysteine-rich proteins that contribute to their water repellent properties. So here in this video, we can observe the difference between a wettable powder formulation containing a wetting agent and one without. So the video features a meta-rhizium strain where the formulation without a surfactant takes over more than 30 minutes to pass the wettability test, while the one with a wetting agent takes only 1 minute to become fully wetted. And there are many WP formulations on the market that lack a surfactant, which can be highly problematic. And without an efficient wetting agent, the product may form agglomerates during dilution in the spray tank, leading to clogging in the application nozzle. So here we can see in these images the difference between a wettable powder with a surfactant and without. And if you don't have an appropriate wetting agent or even a dispersing agent, you can see these the conidia all agglomerated, all clustered. And here in this image, we can see the particles were well distributed, and they do not form these large conidia clusters. As I said, we all know that Bacillus endospores are easier to formulate than fungal spores. But why this happens and why Bacillus endospores are much more resistant to fungal spores? This happens because bacterial endospores, they are real resistance structures. They have this mechanism of endospore forming that evolved as a response of the microorganism to the harsh environmental conditions. And one chemical found in spores, but not in vegetative cells, is dipicolinic acid, which accumulates in the core of the endospores. And this acid, it forms a complex with calcium ions. And this complex of dipicolinic acid and calcium represents 10% of the dry weight of an endospore. And it functions to bind free water within the endospore, thus helping to dehydrate it. And the core of an endospore contains less than 1/4 of the water that we found in vegetative cells. And this dehydration of the core greatly increases the heat resistance of macromolecules within the spore. And they have also small proteins that bind tightly to the DNA structures that will protect it from potential damage from ultraviolet radiation, desiccation, and dry heat. And that's why we can see that endospores are highly capable of surviving at high temperatures like 80 Celsius degrees. And bacillus in an environment that contains water can have a longer shelf life than fungi. And fungal spores, they have more of a reproduction purpose, so they have some resistance to environmental variations, but much less than endospores. They don't have all these apparatus to deal with harsh environments. Another challenge we have is that fungi have been produced by liquid fermentation because solid state fermentation has many drawbacks related to intensive manual handling, cross contamination. It requires huge facilities with controlled temperature and humidity, and so on. So in the future, the majority of fungal strains are going to be produced through liquid fermentation. But generally, it requires a drying downstream to remove all the water from the active ingredient to allow them to be able to survive in the formulations. So we have this challenge to stabilize the fungal structures in the liquid state and protect them from this water available. I'm not gonna deep dive in each one, but different propagules can be formed during this submerged fermentation, obviously depending on the species that you're using. The most common ones are blastospores, submerged conidia, microsclerotia, and chlamydospores. Blastospores are usually more sensitive one, and microsclerotia the more resistant one because microsclerotia they are formed in a response to adverse environmental conditions like low nutrient availability. It's a process that we can relate to the endospore formation. So with more resistance structures that fungi have, and they are dark pigmented due to the presence of melanin that has this property to protect the fungi against UV light. But they have this advantage, but one disadvantage is that when once applied in the field, they tend to have a a, a more difficult action in the field because they need to germinate, form the aerial spore, and then infect the insect. And the blastospores as they are in a more metabolically active way, they can be applied in the field and attack the pest at the same time. To summarize, here we have a scale that shows the general trend in the difficulty of formulating and stabilizing microbial active ingredients. Now, I know that many of you who work in this field might be thinking, oh, I have strains that don't follow this order. And that's true. This isn't set on stone. This scale is based on literature and what we commonly see in scientific articles. However, as I mentioned earlier, the stability depends heavily on the specific strain and the production process involved. So here, as I mentioned, Bacillus endospores are the less difficult to stabilize, and the gram-negative ones are the most difficult ones. Here we have the fungi in the middle, the microsclerotia, they are the most, they are easier to formulate than the fungi blastospores. And then here we have streptomyces, like in the middle of bacillus and fungi. But this can vary depending on the strain and all these particular aspects that I mentioned. So after this first key objective, which is understanding the active ingredient you're working with, the second goal is to ensure we don't harm the microorganisms during formulation. So it is extremely essential to select the right formulation type according to the microbe that you're working. So many common ingredients can be toxic to biologicals depending on their concentration. And that's why it is essential to carefully select and test the right ingredients, the right formulation components. Each component needs to undergo a biocompatibility test to assess how it affects the biological active ingredient and determine at what concentration it may become toxic. In the market, we see a wide range of products with varying levels of quality, and often products containing the same microorganism or even the same strain can differ significantly in quality. And this variation is largely due to the formulation and the choice of the most suitable co-formulants. And some of them don't even has any surfactant in it. So, but now we can see an improvement happening now. Here at Indorama Ventures, we use the minimum inhibitory concentration test to evaluate the compatibility of the bacterial strains with our chemistries to identify the maximum concentrations at which our chemistries can be used safely. This methodology and the equipment we have allow us to test different products at a wide range of concentrations. So we test from 50% to 0.4% and various concentrations between them. And there are different methodologies that can be used to assess this compatibility, but I think that this one is the most reliable one. In using this methodology, we've already evaluated over 30, 40 different chemicals, including individual components, blends, and compositions to offer and recommend the best combinations that won't negatively impact the microorganisms. And for fungi, we use the traditional agar plate method. Fungi are a little bit are a little bit more difficult to perform this test. So we use the traditional one, the traditional method, which is more limited because we can only test fixed concentrations, and it is a little bit time consuming, but we perform this to assess if our chemistries are compatible or not. And with this methodology, we mix our chemistries with the culture media prior to the solidification of the media. And then we place the fungi in the center, in the middle of the agar plate, and then we compare the growth kinetics with the control agar plate. So as a result, we now have our biocompatibility map showing the exact concentrations obtained from our minimum inhibitory concentration experiments with various Bacillus species, a Pseudomonas species, and Trichoderma herzianum. And additionally, we've included a preliminary analysis regarding OMRI eligibility. And as you can see, we offer a diverse range of products suited for various applications. So we have dispersing and co-dispersing agents, wetting agents, emulsifier packages, and adjuvants. And naturally, the selection of each component will depend on the specific formulation you're aiming to develop. And now I'll show you some different ways you can use our portfolio from our compatibility map. So here we have an application case featuring our styrene maleic graphed copolymer. It is a proprietary molecule which demonstrates highly efficient dispersant properties even at low use rates. Here we used a liquid fermentative broth to prepare this formulation, but probably with a solid active, it would perform as well. This polymer offers both steric and charge stabilization of particles, ensuring stability in formulations. It has a high molecular weight and a significant degree of aromatic components in the backbone. So it strongly absorbs on the surface of active ingredients particles, enhancing the overall performance of the formulation. And in our biocompatibility results, we've seen that it had higher biocompatibility compared to the benchmark polymer, the acrylic graphed copolymer. So here we have just some recommendations for emulsifier packages for different liquid carriers. So we have emulsifying packages for vegetable oils, white mineral oils. We have ethoxylated castor oils also. So you can use these different tools depending on the liquid carrier you're using. And what is interesting is that when you use these emulsifying packages in your liquid carrier, it is important to assess the rheology of your formulation. And just a quick observation and discussion I wanted to bring here is that sometimes what we see is that the physical chemical parameters are left aside and the formulator keeps the attention only in the microbial viability. But what we want here is to reinforce that tracking the rheological and all the physical chemical properties of the formulation is extremely important because a good physical chemical aspect will ensure a good tank mix dilution and a application in the field. And understanding the rheology of formulations is critical for optimizing both the manufacturing process and the performance of the final product. And these rheological properties, such as viscosity and flow behavior, they directly influence the stability, shelf life, and ease of application of the formulation. Another handy equipment is lumicizer that can be used to characterize the stability of liquid systems, allowing to obtain insights on quality control, shelf life, and formulation improvements. We use it a lot to verify emulsion stability of oil dispersions. And during a typical lumicizer experiment, you lay down the cuvettes in the equipment. You can place up to 12 samples at the same time, and the equipment apply a centrifugal force to accelerate the separation of the sample. And during the centrifugation, the equipment emits a light that passes through each sample cell, enabling the simultaneous measurement of transmitted light intensity. So we can see the creaming or the sedimentation trends on the formulation and in the emulsion stability. And the software also gives you an stability index. So just to conclude, I like to emphasize that developing a complete and effective formulation requires integrating two key areas of knowledge. So we have the biological aspect of the formulation, which focus on the specificities and viability of the microorganism. And we have the physical chemical aspect, which will ensure that the formulation's properties will deliver the active ingredient in the correct manner. And will ensure that we'll maximize the field efficiency. And achieving this balance, it requires a robust infrastructure, which Indorama Ventures provides. So at our research and development center, we have all the necessary structure, expertise, and capabilities to perform all these analyses discussed today, ensuring that we deliver the best solutions to all of our clients. And that's it. That's what I had to bring today, and I'm open for any questions.
Thank you, Isabella. Obrigado.
De nada.
So, Isabella, while you have control of your slides, we had a question about slide number 22. Could you tell us one more time what greater than 50% means? Get that one out of the way.
We tested the range of the concentration that we tested. The first dilution was 50%. So greater than 50% means that even with 50% of our product, the microorganism it grow normally. So that's why we put the greater than 50%.
Okay, thank you.
Because below of this concentration, they are compatible.
Right. Okay, we do have one more question in the chat, and then 6 more in the Q&A tool. And please, we do have until the top of the hour, so keep these coming. First one was when formulating the wettable powder of fungi, how much is the loading of wetting agents in the formulation? We'll talk about it at the end product here.
Mm-hmm. I think that it depends on obviously on the wetting agent that you're using, but typically it is used around 1% to 5% of wetting agent. And obviously it will depend also in the concentration of the active ingredient, the fungal spores that you're putting in the formulation, because the more you put fungal spores, the more you're gonna need wetting agent to wet these spores. So I think that it depends a lot, but I would say 1 to 5%.
Okay, great.
It's the general concentration.
Another question came in from the chat. Are freeze-dried gram-negative bacteria still metabolically active?
From the papers that we've seen, some of the most part of the biomass loses the viability, but some of them remain active. So I think that depending on the temperature that you use to remove the water, they can remain stable and they can remain active.
Yeah, it's an interesting thing when you talk about the end products, Isabella, because you do need to assay for efficacy for sure, but then from a regulatory standpoint, we know they most certainly are interested in the what metabolites or what bacteria are actually in there. So there are really two aspects of viability in this case. So it's an interesting question. Okay. Going back to the Q&A tool, can you elaborate on why fungal spores are not stable in the presence of water or high water activity media?
They are sensible to water activity because the water triggers the germination of the fungal spores. So, and we only want the fungal to germinate inside the spray tank or in the field. We don't want it to germinate in the formulation. So that's why we don't want water in a formulation that contains a fungi strain.
Right. I'm going to get Minka to clarify what DC is as well. if you could in the Q&A section. Okay. So are these coformulants, Isabella, individually tested for environmental toxicity or just together with the microorganism as the final formula? And I think that answer might even depend geographically as well, but I
Mhm.
might have my theory about that, but I'll let you answer that one.
If the products they have to be tested, I don't know
Right,
if I.
for environmental toxicity. So you have the surfactants individually tested. That list you
No.
showed, those are certainly tested and sent to EPA for testing. But in terms of ecotox, are those tested together in other countries?
I think that it depends on the country
Yeah,
that you are, but some might require this ecotoxicity test. But I don't know if I don't if I understood right the question yet. But the surfactant itself, depending on the application, we have to test for ecotoxicity, biodegradability. We have
right.
to have this information.
Yeah, I mean, from my experience, I could answer for this. I mean, you have co-formulants tested for environmental toxicity, particularly for the U.S. Together with the microorganism, yeah, that's fair game in places like Brazil, for example, at the product level. Uh, uh, where you do have those kind of requirements. And Isabella mentioned biodegradability. That's most certainly is fair game as well in terms of the individual coformulants and the products together. So it does depend on the particular aspect of environmental toxicity we're talking about as well. All right.
Yeah.
Okay. Yeah, thank you, Fernanda. I thought I was right about that one. Okay. Vincent, in the chat, can you comment on colorants that work well with biopesticides in terms of compatibility? I'm imagining that color is very relevant in terms of seed treatment formulations uh, where you do need to add a colorant to make a distinction between untreated and treated seed.
We don't have colorants, if I understood right, in our portfolio. So we don't have any assessment on compatibility of these kind of chemistries. But I'm sure that you must assess if the colorant is biocompatible or not prior to formulating uh, the 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 seed treatment formulation.
Yeah.
But it can be harmful also.
Yeah. Another question from come in. Typically, how long does it take for the MIC method? Does it take for the MIC method to assess the compatibility? Like how long is that test? What's the duration?
It's pretty simple. It takes 48 hours for us to have the results because we perform it in this microplate. So it's 90 cell well plate, and the microorganism it grows inside each plate. So it takes around 48 hours to have the results, and we can test multiple concentrations, multiple products at the same time.
Yep. Is it possible to have a DC formulation of microbes? I know most microbes, it's not soluble in the solvents.
Yeah,
Well, the DC could be in a
yeah,
form of. I know that you could conceivably have it in OD, right? But
yeah. That's why in my slide I put OD/DC
right,
formulation because Some people can say OD, some other people can say DC, because in OD, even in vegetable oil, the fungal spores, they are not soluble in the oil, as it happens in chemical pesticides. So it's, I think all of these would be like disease, actually. Um, so yeah, you can
Yeah,
have a DC, microbial DC.
yeah. Uh, uh, another question, and this goes out to especially you have colleagues in other markets at at Indorama. A an attendee mentioned that there was a manuscript showing that 25% of herbal topical creams and ointments sampled in China fraudulently contained an active ingredient. It's not unusual in the nutrition supplements industry as well. A percentage of supposed organic commodities have also been shown not to be organic. You showed your little chart there on slide number 22, of
Mhm.
course, Isabella. Are you aware of any unlabeled actives being added to biological formulations? There's, I mean, I have my answer, but you can answer this
You
as
can answer.
well. Uh,
I'm not aware. Our own eligibility analysis is just a preliminary one. We don't have, we did not petition for ONRI approval yet. It is going to depend. But we did this analysis regarding based on the matching of cast numbers.
right.
But I'm not aware of.
Yeah, in terms of unlabeled active ingredients, again, we talked a little bit about the regulatory obligations uh, of registrants, both in Brazil, both in the U.S., Europe. This is the case as well. The registrants are obligated to quantify active ingredients to the greatest degree possible, and also provide an enforcement analytical method for these things. So yeah, they and they're supposed to generate a 5-batch analysis that attests to the level of active ingredient as well as any coformulants that might exist. This is why in many places in the globe, we are registering both the active substance as isolated and the formulation as formulated, uh, uh, basically basically testing twice. So and those results are and the products that are produced from that are subject to enforcement matters. So good question there. Uh, okay. We have time for a couple more questions. And I'll tack on a little question on Wendy's here in the question and answer tool. Are the fermentation reactors aerobic or anaerobic? And I want to get the audience to understand where at what point do you get a a uh, a concentrated consistently concentrated active substance to work with when you're scouting for formulations.
So the first question, if it's aerobic or anaerobic, all of these active ingredients, they are aerobic microorganisms. So they need oxygen to grow and to produce their reproductive structures. And here are the bioreactors where the bacillus or the fungi is produced. They have oxygen supplementation during the fermentation process.
Okay. All right. Back to the biocompatibility map. Uh, is there a reason we start at 50% dilution and not any more concentrated than that?
Because most of surfactants, they are not used at that high level of concentration, like 50%. We do perform the tests depending on the application. So depending on the project that we have, we can test higher concentrations. We standardized the 50% because we thought that it would cover the application of most of our surfactants. So that's why we chose this number of concentration. Um, so that's it, but we can test higher ones, and there's no problem.
Great. I think we're out of questions, Isabella.
Hmm.
So I'm going to seize control a little bit to close us out. Thank you very much
Thank you very much.
to you and the audience for participation in this webinar. I'm thanking you, Isabella, for an excellent job, as well as Eurofins Agroscience Services, our sponsor, and our webinar coordinator, Kyle Sawyer. For those questions that we did not get to, please expect answers to these questions to be posted in the next few days. You may be asked to complete a follow-up survey by email after the webinar, and we most certainly would look forward to your feedback. Our next webinar is on November 20th at 12 noon in the East, featuring Dr. Peter Childs, Dr. Gary Patterson, and David Hiltz, who will be leading an interesting panel discussion on the history and future of another biological type product here in the agrochemical world, seaweed-derived products. This webinar will be a joint effort of the Agro and HIST, the History of Science divisions of the American Chemical Society, and we hope will be the start of a series of interesting talks on the history of agroscience and even the American Chemical Society. If you have any ideas for future webinars, please contact me. I'm chairing the webinar committee. And this webinar series is a great opportunity to introduce those outside agro to this dedicated division membership. I'm very happy to introduce Isabella to this audience, and also an opportunity to show the great science of the agro division to a global audience. This is a great opportunity for all of us to build a community. So thank you all for your attention, and we will see you on November 20.